Welcome back, everyone. So, I think you'll be happy to know this is the second video and the last video for chapter 7. So, it's a little bit of a lighter chapter after a slightly stressful week with chapter 5. We're taking a little bit lighter with chapter 7. So, we're just going to talk about how you can evaluate if a disinfectant is good at killing a bacteria or if it's good at stopping the bacteria from growing. We can do something called the disk-diffusion method. In the disk-diffusion method, you take a plate and cover it with bacteria. So here we have 3 different plates with 3 different bacteria. We have a gram-positive bacteria, Staph aureus. This is something that's found a lot on your skin. We have a gram-negative bacteria, E. coli. This would be something that's found in like fecal material. And we have Pseudomonas aeruginosa on this plate. This is a more drug-resistant bacteria, one that can cause infections in like cystic fibrosis patients. So in certain situations, this is a particular concern. So after you coat your plate with your bacteria, you take these little paper discs and you soak them in your chemical, whatever your disinfectant you're studying. Here we're studying chlorine, hexachlorophene, O-phenylphenol, and a quaternary compound. You just dip your paper disc in the disinfectant and then place it on top of the bacteria. And you incubate it overnight. And then sometimes you'll see this zone of inhibition, this halo around the disc. This tells you that this disc, the disinfectant on it, stopped the bacteria from growing around it. Here you've got a zone of inhibition and a zone and a zone. You can measure that zone and figure out how effective that disinfectant is at preventing the growth of that bacteria. So here if you're trying to clean like maybe a cell phone or something that gets touched a lot, Staphylococcus aureus would be killed by any of these. So any of those would be a decent disinfectant. If you're trying to kill E. coli, chlorine, bleach would be pretty good, but hexachlorophene, see how there's no halo around it? There's no zone. Hexachlorophene did not kill that bacteria. So you would say that is not a good disinfectant if I am cleaning a toilet or something. Then for Pseudomonas aeruginosa, all 3 of these were unable to prevent the growth of bacteria. So these would not be a good disinfectant if I'm trying to kill this bacteria. But chlorine does a little bit of inhibition of growth. So that would be able to slightly inhibit that bacteria from growing. Okay, so a question. If you want to disinfect a surface contaminated by vomit and a surface contaminated by a sneeze, would your choice of disinfectant make a difference? Let's talk about some chemical methods to control bacteria growth. Phenols will disrupt a plasma membrane, so something like Lysol. Biguanides will disrupt plasma membrane, something like chlorhexidine that's in topical antiseptics. Alcohols are able to both disrupt plasma membranes and denature proteins. These are good at, cleaning off a table before lab. These are not good antiseptics. You do not want to put alcohol on a wound because it'll actually start to coagulate the protein on that wound and give a little space for the bacteria to grow underneath. Ethanol, isopropanol, ethanol being hand sanitizer like Purell, and isopropanol being rubbing alcohol. Heavy metals, things like copper, zinc, mercury, these can denature proteins. So they're often found in things like mouthwashes, anti-dandruff shampoo. So, it'll say there's copper in this, there's zinc in this. Burn creams will often have heavy metals in it. It only takes a tiny amount of a metal to kill a microbe. This is called oligodynamic action. Here's a plate covered with bacteria. That's what this yellow creamy color is. And here you can see they put different coins, different metal things on it. And when they remove the little piece of metal, you can see the metal actually inhibited that bacteria from growing. A word about silver, though. Sometimes people will tell you that they take silver to, ingest it to stop microbes from growing. Not something I recommend. It can permanently turn your skin blue. So while silver can kill microbes, not a great thing to be consuming regularly. Halogens, things like iodine and chlorine, these are things that are on the next to last column of the periodic table. Iodine can stop protein synthesis, can alter membranes, something like betadine. Chlorine can oxidize, something like chlorine bleach. Surfactants are things that are able to decrease surface tension. So something like a soap that's used to degerm and mechanically remove. And quaternary ammonias, which can denature proteins. Chemical food preservatives are chemicals that are added to food to stop them from spoiling. So you might read things like sorbic acid, benzoic acid, calcium propionate. They stop your food from molding. They stop bacteria from growing. Nitrite can also be added to stop botulism into spores from growing. So you're not having growing botulism, leaving botulism toxin. If you can just tell me a chemical food preservative is added to food to preserve it, you don't need to be able to tell me this list. That's a little excessive. Aldehydes can inactivate proteins. These are some of the most effective antimicrobials. So something like formaldehyde gas or formalin. Used to be, this is how biological specimens were preserved. So you'd have that nice smell of a biology lab. Antibiotics. There's also antifungals, anti-endospores, different chemicals, things like niacin, netamycin. And then fumigation are gaseous sterilants, something like chlorine dioxide. This does require a closed chamber so that gas doesn't escape. But this can be used to remove a particular species. Plasma, I think of plasma as like the hot part of a flame. The hot part of a flame. This is when the gas is excited by an electromagnetic field. And you get free radicals. And those free radicals are what are really good at destroying endospores. And then peroxygens, which can oxidize. Things like hydrogen peroxide, benzoyl peroxide, and ozone. Hydrogen peroxide used to disinfect non-living surfaces. Benzoyl peroxide, which is an acne medication because it kills bacteria that can cause acne. And then ozone, which is used to disinfect water. Ozone doesn't have a particular taste or odor, so it's really good at disinfecting water because water still remains potable. And then bacteriocins, which are proteins that are produced by one bacteria that inhibit another. Totally crazy things. Like niacin and netamycin, created by one species of bacteria that creates the cheese and prevents other bacteria from spoiling the cheese. It gives some bacteria kind of a beneficial. They're not having as many bacteria competing around them for the space, so it's not uncommon for some microbes to produce antibiotics that kill other microbes. Okay, then we need to also consider the microbial characteristics. So the easiest to kill, least resistant microbes would be things like viruses with lipid envelopes. Just destroy the envelope. It's easy to destroy those. Gram-positive bacteria, destroy that thick cell wall. They lyse immediately. Prions. I don't know anything that can effectively destroy a prion. Prions are misfolded proteins. We talked about these the start of the semester. Not really much you can do except for just avoid them. Endospores of bacteria. Endospores are created specifically with the goal of being resistant. So no surprise, they are some of the most resistant characteristics that a microbe could have. Okay, so a couple of questions. Why would alcohol be effective against some viruses and not others? A hint, I would look here. There are some viruses here, some viruses here. What chemicals can be used to sterilize? Okay, that's it for Chapter 7. We'll pick up with Chapter 8. We'll cover Chapter 8 and Chapter 20 before your second exam. So you're about halfway there. I'll see you in our next video. Bye.